Myoglobin in muscle food. Myoglobin is a cytoplasmic protein which is water-soluble and determines milk color. It is made of 8 alpha helix with a Pacific heme group, contains a center ion atom. The ion atom has 6 binding sites. 4 of the binding sites connect to the heme ring with 4 atoms. The 5th site attached to the proximal histone, and the 6th binding site binds to ligands such as divalent oxygen, water, and carbon monoxide. The state of oxidation and oxygenation of the various ions in the Pacific heme group determines milk color. The 4 chemical forms of myoglobin which are responsible for milk color are deoxymyoglobin, oxymyoglobin, carboxymyoglobin, and metmyoglobin. Deoxymyoglobin is reduced myoglobin. Its pigment is in purple-red color. The ion atom which in the center of Pacific heme group is reduced to ferrous ion state, and no ligand connect to the 6 coordination site. It is usually in the center of milk where oxygen is not present. To maintain deoxymyoglobin needs very low oxygen in the center of the milk. When the 6 coordination site attached to carbon monoxide with ferrous ion located in the center of the Pacific heme group, it forms carboxymyoglobin. It gives a stable bright red color of milk when the oxygen tension is very low. Oxymyoglobin, also called bromine, is oxygenated form. The oxymyoglobin gives a desired bright red color in fresh milk. Gravelin oxygen is attached to the 6 coordination site of ferrous ion. Oxymyoglobin is formed on the surface of the milk and need high oxygen tension. Brown color myoglobin forms in the middle of purple-red layer and the bright red milk layer. Where the oxygen tension is low, myoglobin is oxidized to myoglobin. Ferrous ion become ferric ion, with the water as the ligand attached to the 6 binding site. Reaction 1 of the milk color triangle is depending on time, temperature, pH, and the composition of oxygen by mitochondria and the level of oxygen penetration. Reaction 2 is unlikely to happen because oxymyoglobin inhibits metmyoglobin formation. Reaction 3 needs a low oxygen partial pressure. When the partial pressure of oxygen is high, the oxymyoglobin will oxidize to oxymyoglobin. Reaction 4 occurs due to enzymic and non-enzymic applications. Reaction 5 happens when the oxymyoglobin contact with carbon monoxide. This reaction is reversible reaction but favors the forward reaction.
There are a number of mechanisms operating in muscle foods which have an influence on myoglobin oxidation. One of them is autooxidation, which is usually favored at higher oxygen levels. Another factor which affects is high temperature, because at high temperature the rate of dissociation of oxygen increases, which increases the tendency of deoxymyoglobin to oxidation. And it has been shown that myoglobin with high oxygen dissociation rate have higher tendency of oxidation. Under aerobic conditions, spontaneous autooxidation is enhanced by various metal salts and complexes like divalent cupric compounds, which are naturally present in biological structures. Binding of copper, especially at inner histidines at position 97 and 64, alters the conformation and stability of myoglobin, eventually increasing the rate of oxidation. The partial pressure of oxygen plays a central role in all of these activities, which is lowered by microbial metabolism, mitochondrial activity, and lipid oxidation. In presence of oxygen, as could be the case with MAP packaging and catalysts like non-heme or heme iron, unsaturated fatty acids in the cell wall gets oxidized, and this secondary breakdown products are principally involved in oxidation of myoglobin to form metmyoglobin. One of them is 4-hydroxynonenal. Also, with the decrease in dissolved oxygen level due to lipid oxidation, it again favors oxidation of myoglobin. Another important factor is oxygen consumption rate, which varies among different muscles of the same animal species. It is principally attributed to different kinds of muscle fibers. For example, in psoas major, which is rich in type 1 fiber, which have higher myoglobin content and higher mitochondrial concentration. At higher mitochondrial concentration, which favors higher respiration rate and eventually leads to decreased oxygen partial pressure in the local muscle environment. This leads to formation of shallow myoglobin layer on the surface, and also it favors formation of deoxymyoglobin rather than oxymyoglobin. And deoxymyoglobin is less stable compared to oxy1, and it eventually gets oxidized to brown matte myoglobin. On the other hand, muscles like longissimus dorsi, which has lower oxygen consumption rate, are much more color stable. Completely different mechanism is governed by MAT myoglobin reducing system. It is attributed to NADH cytochrome B5 reductase, which is present in mitochondria as well as sarcoplasmic reticulum. At mitochondria, it utilizes outer mitochondrial membrane cytochrome B as an electron transfer mediator, where it is converted from oxidized to reduced form, which in turn reduces MAT myoglobin to myoglobin. While as seen in part B at sarcoplasmic reticulum, the electron transfer mediator is cytochrome B5, the reduced form of which again converts MAT myoglobin to myoglobin. In both of these cases, enzyme utilizes NADH as coenzyme and electron carrier, and thus the reducing ability is completely dependent on NADH pool. Another enzyme having contribution in MAT myoglobin reducing system is lactate dehydrogenase. It generates NADH by catalyzing reversal of electron transport, where it oxidizes lactate back to pyrite with simultaneous formation of NADH, which is utilized by MMB reducing system in conversion of ferric to ferrous myoglobin, and thus maintaining the color stability of muscles. But with postmortem time, NADH pool decreases and eventually the MMB activity, and also MMB activity is negatively affected by high O2 levels. Thus it is complex interplay of various mechanisms which ultimately decides the oxidation of myoglobin in muscle foods.
The oxidation states of myoglobin are influenced by packaging methods and oxidizing agents, such as in cured meat. Raw beef steaks display higher values of redness when packaged in modified atmosphere packaging with 70% oxygen, 30% carbon dioxide, compared to 30% oxygen and 70% carbon dioxide. At high oxygen levels, the dominant redox form of myoglobin is the ferrous bright red oxymyoglobin. The decrease in redness in the low oxygen modified atmosphere packaging and the change in colour from red to brown is attributed to the formation of the oxidized ferric myoglobin. In high oxygen atmospheres, lipid oxidation occurs more readily, and byproducts catalyze the oxidation of oxymyoglobin to metmyoglobin. Thus, it is a trade-off between the fresh red meat colour and the formation of lipid oxidation products, leading to the subsequent myoglobin oxidation. Alternatively, meat can be packaged without oxygen. Vacuum packing alongside oxygen impermeable material and oxygen scavengers ensures that partial oxygen levels are low. In these conditions, the formation of the purple-red ferrous deoxymyoglobin is favoured over the red oxymyoglobin. When the ferrous forms of myoglobin are heated, the globin protein is denatured and the heme group, which was protected by the hydrophobic lens, becomes exposed and susceptible to oxidation. This results in the formation of the red globin ferrohemochrome, which in turn becomes oxidized to the grey-brown ferrohemochrome. However, if oxidation does occur prior to cooking, the globin protein of ferric metmyoglobin is denatured, producing the grey-brown ferrohemochrome with no intermediate step. The ferrous forms of myoglobin are more heat-stable than ferric metmyoglobin, which accounts for the rapid formation of ferrohemochrome, a process known as premature browning. This raises consumer safety concerns as the meat often looks cooked before it has reached a safe cooking temperature. Oxidation of myoglobin can also occur through the action of nitrates such as sodium and potassium nitrite, which are common additives used in cured meat products. Nitrates are highly reactive compounds that oxidize myoglobin from ferrous to ferric metmyoglobin through a series of reactions as shown. In an acidic environment, such as meat with a pH of around 5.5, nitrous acid is formed from nitrite. Its anhydride nitric oxide forms a bright pink-red stable complex with myoglobin, nitrosamyoglobin. When this compound is heated, the protein moiety becomes denatured, but the red nitric oxide porphyrin ring structure is preserved, retaining the characteristic cured pink-red color known as nitric oxide hemochrome.
Factors affect the oxidation of myoglobin, which in turn affects the quality of the meat. pH. It is observed that a postmortem skeleton muscle pH in beef, pork, and lamb is typically 5.5 to 5.8, whereas pH of avian muscle range from 5.7 to 6. However, myoglobin is quite stable at 7.4. Breaking of glycogen occurs as the muscle is converted to meat, and this results in accumulation of lactic acid, which in turn decreases the pH. This decline in pH promotes oxidation of myoglobin as the globin moiety undergoes denaturation and consequently oxygen molecule dissociates from the heme group. As acids are well-known oxidizing agents, they bring about the oxidation of myoglobin to metmyoglobin. The rate of oxidation increases as the pH decreases. A condition known as PAC, which stands for pale, soft, and exudative, which occurs in pork, beef, and poultry, is caused due to the low pH, which results in oxidation of myoglobin to form metmyoglobin that has a low color intensity. Another factor that majorly affects the oxidation of myoglobin is light, which has been reported in many studies. A study of photosensitivity of fresh meat in visible spectrum revealed that there is no specific wavelength where the oxidation of myoglobin is enhanced. However, 5.5% of oxidation was observed in myoglobin of exposed sample. It was then justified that a photochemical compound like riboflavin reacts with oxymyoglobin, thereby increasing the oxidation rate to form metmyoglobin. In terms of radiant energy, an increase in metmyoglobin content was observed in synthetase muscle. It is hypothesized that the radiant energy leads to oxidation of lipids, which further leads to increase in the pigment oxidation, thereby increasing the metmyoglobin. With respect to fluorescent light, a slight acceleration in oxidation was observed in bovine, porcine, and oven muscle, while no effect on oxidation were observed in presence of long wavelength red fluorescent and incandescent light. High temperature promotes myoglobin oxidation. At high temperature, function responsible for protection of heme group of globin moiety is lost. As a result, oxymyoglobin is deoxygenated to unstable reduced myoglobin. As the myoglobin is very unstable and the heme protein is relatively unprotected, oxidation occurs spontaneously to metmyoglobin. Oxygen utilizing enzyme activity present in the muscle actually brings about the oxidation. Therefore, as the temperature increases, activity of this enzyme